Hello students, let us see how we prepare the tissue culture media using the different types of components in this video session. We will also learn how generally the selection of a suitable plant tissue culture media is done for the different types of plant species as well as explants. As we all know, plant tissue culture is the science or art of growing plant cells, tissues or organs isolated from the mother plant in an artificial media under controlled environmental conditions. The plant tissue culture relies on the concept of totipotency, that is, many plant cells have the ability to regenerate into a whole plant when they are provided with required nutrition and environmental conditions. The explants or the initiating material could be single cells, protoplast, pieces of leaves, stems or roots, or even floral and reproductive organs. The exact conditions required to initiate and sustain plant cells in culture or to regenerate intact plants from these cultured cells are different for each plant species. Each variety of a species will have a specific set of cultural requirements, one among which is the media. 5 widely used culture media are described in this session briefly. Murashige and Skoog medium was invented by two scientists, namely Toshio Murashige and K. Skoog, way back in 1962. While these two scientists were working on the discovery of plant growth regulators, nowadays it is the most commonly used medium in tissue culture. Sometimes there are numbers indicated following the term MS, which generally indicates the amount of sucrose that is present in the medium. For example, MS10 indicates the presence of 10 grams per liter of sucrose in the media. For most wide purposes, 30 grams of sucrose per liter of MS media is generally used. Murashige and Skoog medium is suitable for a vast range of plants and explants. They are generally used to induce organogenesis, callus culture, micropropagation as well as in cell suspension cultures. LS media or Linzmeyer and Skoog medium. The Linzmeyer and Skoog medium was developed by two scientists, namely Linzmeyer and Skoog, in 1965. They had developed this media first to optimize the organic supplementation in tobacco culture. The media has a more or less similar composition of Murashige and Skoog medium, except for the difference in the concentration of vitamins. Certain vitamins like thiamine hypochlorite are more in LS media, which compensate for the absence of inositol in this media. LS media is preferred for organogenesis, callus culture, cell suspension culture as well as micropropagation of plant species. The B5 media developed by O.L. Gamborg was first used for the callus induction and cell suspension culture of glycine max and the other plants belonging to the family Fabaceae. This media is a blend of several inorganic salts, vitamins and carbohydrates like any other tissue culture media. The unique aspect of this Gamborg medium is it has a higher concentration of nitrate and potassium and a lower concentration of ammonia compared to other tissue culture media. This was formulated in such a manner because potassium nitrate was found to be useful in inducing the root callus formation in plants like soybean and ammonium sulfate an essential role in cell growth. The major purpose of Gamborg media is protoplast culture. The N&N media developed by Niche was originally developed for establishment of anther cultures in vitro from plants like Nicotiana belonging to the family Solanaceae. It has a high concentration of thiamine, biotin and folic acid to support anther culture. White's medium was developed by P.R. White in 1963. It has a specific purpose of shoot culture and callus culture. This was one of the earliest plant tissue culture media developed. It has a low salt concentration, and higher concentration of magnesium sulfate compared to other plant tissue culture formulations. It is generally preferred for culture of Musa and Daucus species. Moving on, media selection is a very crucial step in tissue culture. There is no single step to formulate a suitable media for the establishment of a culture of any plant species. It may take several years to develop a suitable tissue culture media, which vary with the species as well as the explant, which means within the same plant variety or species, for the use or culture of different explants, that is the shoot tip or the leaf or any other organ, the media composition may have to be varied. So people try different combinations of inorganic salts, organic supplements and plant growth regulators to develop a medium which can promote regeneration from a particular species of plant and explant. Generally, this trial and error method has three major combinations that are varied, that is the salt concentrations, then different proportions of auxins and cytokinins, and different concentration of sucrose, which may vary between 2 to 6%. The arrangement of all these numerous components of media require a specialized area in the tissue culture laboratory, which generally we call as a media room, which has to be maintained at high cleanliness levels without any contamination that can lead to spoilage of cultures. Generally, media rooms are maintained with restricted entry of people. Media needs to be prepared with utmost care and following only the recommended procedures without deviations, so as to lead to successful tissue culture process. The water that is used for in media preparation needs to be distilled water, and many times sterilized water also is preferred for media preparation. Media preparation is the major step which is very important and which is the first step in tissue culture process. All the different components mentioned in the composition of the tissue culture media are weighed accordingly and then dissolved in various steps so that they all undergo proper dissolution. Then the media is allowed for jellification and then for sterilization. Weighing all the numerous components in a tissue culture media generally will consume a lot of time and effort. Also, many of these elements or components are required in very minute or even trace quantities. Weighing such small quantities of substances becomes very difficult. So, accuracy cannot be maintained. To overcome these difficulties, generally concentrated stock solutions of tissue culture media components are prepared and stored. So, a stock solution is a solution of components which is at a very high concentration than what is actually required in the usable or working media. So, this is a very convenient method for doing plant tissue culture. So, these concentrated stock solutions are then diluted as when required to the preferred concentration of the working solution before use. The stock solutions are stored in refrigerator generally at 2 to 4 degrees. Some of the components, that's for example stock solutions of micronutrients or even vitamins usually are stored at -20 also if they need to be stored for a longer time to preserve their stability. Generally, macronutrients are prepared as stock solutions which are 10 times the strength of final operative medium. Micronutrients are made up to 100 times of the final concentration in the working medium. Iron stock solution generally is prepared separately as a chelated solution of iron EDTA. Otherwise, the presence of iron in the media will interfere with the absorption of other nutrients in the media by the plant. As such, iron is prepared as a chelated solution using EDTA. So, the FeEDTA allows slow release of iron into the tissue culture media as and when required. So, as the concentration of iron keeps on decreasing in the media due to absorption and usage by the growing tissues, more iron is released into the media. Vitamins are prepared either 100 times more than the required concentration or sometimes even 1000 times more than the required concentration. So, the prepared vitamin stock solutions are stored at minus 20 degrees if they have to be stored for a longer time. Plant growth hormones are also prepared at a higher concentration, generally 1000 times than the final desired concentration. The concentrations of inorganic and organic components of the media are generally expressed in milligrams per liter of the media or as parts per million, that is ppm. The International Association for Plant Physiology recommends the use of mole values to describe the concentration of different components in the plant tissue culture media. So, once the stock solutions are prepared and stored, the desired concentration or the desired volume of stock that has to be used is calculated using these two formulae. So, the desired concentration of stock solution is equal to the amount of stock solution taken for 1 liter media. If the quantity of medium that is to be prepared, that is a working medium, is less than 1 liter, then the required concentration or required volume of stock solution is calculated by using the second formula as shown here. So, for an example, I have mentioned the volumes of different stock solutions, namely macro, micronutrients, then potassium iodide, which is a source of potassium, then stock solution of iron EDTA, then the 3 different vitamins, stock solution of amino acid supplement, that is glycine, and mesoinositol or myoinositol for preparation of 1 liter of Murashige and Skoog medium in this table. So, practically while preparing 1 liter of MS media, first 30 grams of sucrose is dissolved in 200 ml of double distilled water. If recommended, 1 to 2 grams of activated charcoal is weighed and added to the solution. Generally, the activated charcoal solution is prepared and filtered using a filter paper and suction pump. The remaining stock solutions are measured in volumes as given in the table and added to double distilled or sterile water in another container in the same sequence to make it up to around 700 ml in volume. To this, a filtered sucrose solution is added and the mixture is taken into a 1 liter measuring cylinder or any other measuring container. Then the pH of the liquid medium is adjusted to 5.6 to 5.8 with the help of 0.1 normal hydrochloride or 0.1 normal NaOH. The operation is done generally preferably using a pH meter. Once the pH is brought about to 5.6 to 5.8, then the final volume is made to 1 liter using double distilled water. The entire contents are mixed well to make a uniform or homogeneous solution. To this solution, agar is added at a concentration of 0.8 to 1% and heated to 60 degrees to allow the melting of agar completely. On cooling, this would give rise to a semi-solid media. In case we are going to use liquid media, then agar is omitted. So, the prepared solution is then dispensed into culture containers, maybe test tubes or tissue culture bottles or any other required containers, and these are then closed with non-absorbent cotton plex and then a layer of gauze cloth is applied. If plastic lids are available for the culture containers, they are applied but not very tightly. They are allowed or fitted a little loosely Then these containers are finally sterilized by autoclaving. So this is how the different types of media are prepared and made ready for tissue culture process. You can refer to these references. Thank you.